So welcome to Biomanscape. Today we are going to know about the Western blotting. It is also known as immunobloting and it is a laboratory technique used to detect specific protein in a sample. So Western blotting is a method in which the protein are first separated by the size using the SDS-PAGE and then that is a gel electrophoresis by that and then transferred to a membrane and finally identified using a specific antibody. So why it is called Western blotting? The name comes from an analog with Southern blotting used to detect the DNA and it was developed by the Edwin Southern. So later the technique was named according to the Southern blotting, it was used to detect the DNA, whereas Northern blotting is used to detect an RNA, whereas the Western blotting it is used to detect the protein. So it combined two things. The Western blotting it separates the protein by electrophoresis and by antibodies it specify the detection. So the applications of Western blotting are like detection of diseases related to the protein, example HIV, Lyme disease or prion disease. Then it checks whether the protein expression is in researches and it verify the result for other tests like ELISA confirmation. And for studying the post-translation modification, for that also it is used. So Western blotting is a protein detecting method that uses the antibodies after gel separation. So let's know the steps of Western blotting directly. So first is the protein separation by SDS-PAGE. So the protein from the sample are first denatured and coated with SDS, a detergent, to give them a uniform negative charge. Then they are then separated on the polyacrylamide gel SDS-PAGE according to the molecular weight. That is, the small protein move faster. Then transfer to the membrane. The separated protein on the gel are bloated onto the nitrocellulose or PVDF membrane using an electric field. This step makes protein more stable and accessible for the antibody binding. Then blocking, important step to prevent the non-specific antibody binding the membrane in an incubated with a blocking solution, like example protein, milk or BSA. So this converts all the empty spaces on the membrane. Then primary antibody incubation, the membrane is then incubated with the primary antibody like the patent's antiserum and which specifically binds to the targeted protein, that is antigen. Then secondary antibody incubation, next the membrane is incubated with the secondary antibody that is conjugated with an enzyme like a horse radish peroxidase or alkaline phosphatase. So the second antibody bind to the primary antibody and this amplifies the signal, and since multiple secondary antibody can bind to one primary antibody. So next step is detection. A substance of the enzyme is added and the enzyme convert the substrate into a colored or luminescence product. So this reveals the presence of the protein as a band on the membrane. So the band correspond to the protein of specific molecular weight, and the presence or absence or intensity of the band provide qualitative and sometimes semi-quantitative information about the protein. So let's know like a protocol for that, like protein separation by SDS-PAGE, then bloating to nitrocellulose membrane, then blocking membrane, that is to prevent the background noise, and then incubate with the primary antibody, then finally incubate with the enzyme linked secondary antibody, then add substrate, that is enzyme product detectable signals, and then band appear, that is analyze vision is available. So the separation is by SDS-PAGE, bloating is transferred to the membrane, T that is blocked and prevents non-specific binding, antibody 1 it is primarily involved, and then binding of antibody 2 secondary with the enzyme, and detection that is from substrate to finally the visible bands are observed. Thanks for watching. Do like, share and subscribe.